be able to give this talk. And the inspiration for the title, actually, I was trying to figure out, I hate coming up with titles for things. So I was looking at my bulletin board, and I have all of the buttons that the paleobotany section produces. And one of the early ones is Dead Plants Tell No Lies. And I thought, that's pretty good. So I'm going to borrow that. Because the paleobotany section of BSA has been a real strong support, and I don't think I'd be here without everyone in that section. So it's kind of my homage. And just to name a few specific people who've had a really big influence on me, this includes Ruth, my PhD supervisor, and Margaret, my postdoc advisor, Gar, who I guess is kind of a de facto advisor. Tom Taylor, who unfortunately passed away a little bit ago, as most of you know. He was such a huge influence in paleobotany, so it's a real loss to the community. My lab mate, Stefan, as well as two of my current collaborators, Chelsea, who gave the very nice introduction, and Steve Manchester. So, and I'd like to, all of my students that I've been working with at Michigan, past and present. So, this, obviously what I'm talking about isn't just my work. There's a lot of people involved, so I'll try and highlight them as we go through. So, so as Chelsea said, I started with Ruth in undergrad, but it was actually in high school that I started working with her. And I'll admit, I've had an obsession with fossils for a very long time, since grade 3. And I did go through a phase of liking dinosaurs, but I came to my senses in high school. And I think I really like this image because it says it all, right? Here are the dinosaurs. That's about how important they are, right? The plants are what's really important. And for a paleobotanist, we have a bunch of different reasons why plants are important. They're the primary producers in terrestrial ecosystems, so they're this foundation. So in order to understand what kind of environment the animals are living in, what's happening on land, we need to know what the plants are doing. They're also, because they're stationary, they really reflect the environment, they interact with and shape the environment. So if we're trying to understand how things have changed over geologic time, we need to know what the plants are doing. And what's more relevant for this crowd is that we can see different ways that evolution happens, right? Plants don't work the same way as animals, so they can give us different insights. They do things differently and in more interesting ways. So why do we need fossils? So when we have fossils, this tells us something about evolution, right? We can put this in a phylogenetic context. We can look at character evolution, that kind of thing. This also tells us simply about biodiversity, so how many species have there been, how many species in specific clades. We can also use fossils to reconstruct biomes and ecosystems, so this gives us the slightly higher level systems, and we can again see how that's changed through time. And then we can understand how both individual lineages and biomes and ecosystems respond to global change over geologic time, which is becoming, I think, quite important as we move into more an era of global climate change with anthropogenic warming. In order to predict what's going to happen in the future, this is what's going to tell us what's happened in the past and how plants and ecosystems have dealt with these kinds of changes in the past, right? We have natural experiments here that are preserved in the rock record to help us understand that. And so fossils provide evidence for novel taxa, novel characters, and novel distributions that cannot be inferred or predicted by just looking at molecular modern evidence, right? So, and I'll show you a few examples of that. And so in order to get the most out of your fossils, there's a certain amount you can do without naming it, but really a core of what I do, what a lot of us paleobotanists do, is figuring out the name, and along with that process, we can figure out some of these other things. So you all know this quote from Shakespeare: "What's in a name? That which we call a rose, by any other name would smell as sweet. But which rose are you talking about?" Right? So it makes a difference for what you're going to interpret, which rose you're talking about. So we need to be confident in what our fossils are, so that the whole process of identifying them is going to help us put them in the correct taxonomic hierarchy. So that's going to affect inferences of phylogeny and evolution, biogeographic reconstructions, understanding the timing of the origin and diversification of different groups, and also looking at ecological or environmental characters, where we're inferring them based on what their modern relatives are doing. So that all comes down to this process of identifying the fossil. And because we don't have DNA, a lot of that relies on looking at morphology and anatomy. So this is a little diagram Chelsea and I came up with that I think really illustrates the benefits of combining morphology, ecology, and genetic data in order to really understand the complete history of a group. It doesn't matter which group it is. So a lot of my work is motivated by this kind of stuff. So how do fossils compare to the modern plants? So we can look at anatomy and morphology, we can look at diversity, ecology, and evolution. When and where did different groups originate and diversify, and what were the drivers of those and the consequences? And then how can we use fossils to reconstruct past environments? So most of this talk is just going to focus on this first part, but I've got a couple of short examples for the other two. So I've worked on a bunch of different plant groups, and they all have their interesting points. I wish I had more time to work on more of them. But one I've been spending a lot of time with are the monocots, so I'm going to focus on that for this talk. So monocots are a monophyletic group. They're about 22%, so almost a quarter of angiosperm diversity. These include a lot of common plants, right? You guys all know monocots, things like lilies, orchids, grasses, and palms. Monocots are economically important, so this is one reason why it might be worth understanding their evolution and response to global changes, right? A lot of our staple plants are monocots, things like grains and bananas, plantains. We also use them for fiber and construction, and we have a lot of ornamental plants, right? So there are a bunch of different reasons why people care about monocots. They're also very ecologically important, so they form the dominant parts or very characteristic parts of numerous different ecosystems. And so this is part of how we can use them to reconstruct past environments, things like grasslands, the fynbos, through the palm savanna, the seagrasses, right? The only marine angiosperms are in the monocots. A lot of wetland environments are dominated by monocots. And then here's a nice tropical forest showing a lot of gingers and palms that are very characteristic understory plants there. So one of our challenges as paleobotanists are, unlike neobotanists, we don't tend to get whole plants. So we love it when we get whole plants, but usually we're dealing with just individual parts, right? And we have to identify those and put them into context without having the benefit of the whole plant, unless we're really lucky and have exceptional preservation. So this can be a little bit of a problem, kind of like the blind man and the elephant, right? Depending which part we have, we might infer different things. So reproductive structures can be really useful because they tend to have a lot of characters. So since we're talking about angiosperms, that's fruits and seeds here, a lot of systematically useful characters. One of our other challenges though, can be dealing with differential preservation of these organs. So where we get combinations of hard and soft parts, this may decay at different rates, or different parts can get infilled with sediment, and we might get just a cast of the inside of the fruit or seed that gets preserved. And so this can be challenging across different kinds of fossils and across different localities if we're trying to understand what the diversity, actual diversity is across a landscape. So here you can see a walnut, right? And so a walnut has this fleshy outer husk, and if you remove that, so it's just the endocarp, that looks quite different. And then if you look inside the endocarp at the seeds, we have the cotyledons filling that, and those also have difference. So if you imagine finding these as 3 different fossils without something exceptionally preserved that has all of them, you might call these 3 different species instead of it being 1 species. So this is something we need to keep in mind for how can we address that. And so one of the methods that we've been using, what can help address that and help address other things, is X-ray tomography. This is a really fun technique to do. If you're not familiar with it, just very briefly, what we do is we take a specimen, so this is millimeters here, and we glue them onto toothpicks or little stubs. They go on a stage, and then as that stage rotates 360 degrees, we get X-rays going through it, and a camera takes a series of radiographic images. So this is just like an X-ray of your hand. And then there are computer algorithms that take that whole series of images and change them to the computed tomograms. And so here we get cellular level details. We get this big stack of CT images. And so we can read these into a 3D visualization program and analyze it. So this is what we call a volume rendering of this seed. So there are a couple different ways. I'm not going to go through this in much detail, but there are desktop CTs, micro CTs that are pretty common, or more commonly available, and then there are synchrotron-based methods, and we're using both. So there are positives and negatives with each of them. So just to give you an idea, this is through a banana seed. So you can see this is a longitudinal section through the whole seed. And here's that volume rendering, so you can see the embryo, for example, right here, this operculum structure. So they're really fun to look at. One of the nice things with them too is there are a lot of benefits. One is that it's non-destructive. So we can actually look at very rare, very fragile specimens, which is really helpful, especially for our fossils, where museums and collections usually don't want things destroyed, especially if there's only one or a few of them. So this way we can still get that internal structure without damaging the specimen. But equally for modern plants, right, fruits and seeds aren't often collected or aren't always available in herbaria. So again, this is a way we can borrow it, get the info, but then the seed can go back to the herbarium and it's still intact. It's fast, so these take somewhere between maybe 10 minutes to an hour, depending on the size of your specimen and your resolution. They're high resolution, so we can get, again, it depends a bit on the system, but quite commonly something like 0.2 microns. So we can see all of the cell details, or cellular level details. So it's quite useful on difficult to prepare materials, so things that have a combination of hard and soft tissues or a lot of silica in them, that kind of stuff. And we get both internal and external data. And so we can section these. So this is a many sperm fruit. We can get sections through one data set in any plane of section we want. This can be nice for our fossils. We don't always get to control which plane of section that they're in. And so this way we can take a modern comparative one and look through it at weird planes of section and see if we get the same kind of structures appearing. We can also virtually dissect or infill structures. So here's that same fruit. So that's the whole fruit. If we remove this outer fleshy layer here to reveal the endocarp, you can see that's quite different. This is the seed inside, and then this blue one is a virtual cast of the space inside the endocarp. So what would happen if that endocarp got filled with sediment, and then that's all that got preserved. And this, for many sperms, we get fossils that span these ranges of preservation. So this kind of thing could, again, help us to better compare across localities where we have some preserved as loculus casts or some preserved as endocarps to try and better match up the morphologies. So I just want to show a couple examples of some work that we did with some fossils from the Mussel World Heritage Site in Germany. This is a Middle Eocene site. It's 47 million years. You can see it there. So these are oil shales. So it's a site of very exceptional preservation, and it's quite famous for its animals. Things like the beetles still have their iridescence on them, but the plants are quite exquisite as well. And so the first fossils we were looking at started coming about because of this fossil seed that's found in the UK that was placed in the sedge genus Scirpus, so an extant genus. And then that same seed morphology was found in these fruits. And you guys are all botanists, so I'm sure you can recognize this is not a sedge fruit. And so we were going, what kind of fruit is that? And what we figured out was that it matches modern Cyclanthus, which is in the Cyclanthaceae and the Pendinales. So these discs are a whole bunch of fused pistillate flowers, and they fall off the infructescence axis when they're mature and then split open to reveal all the seeds. And so here's one of the modern seeds, and you can say, okay, well, the fruits match, but the seeds don't look at all similar, right? So we thought, well, maybe something's happening chemically in how these are getting preserved that's altered the shape of the seed. And so we scanned the seed. So a whole seed is about 0.5 millimeter in diameter. So these are not something that you could do mechanically very easily to remove these layers. And let's see if I can replay the movie. But we did torture a few with nitric acid, since this was the first time we did it, just to mimic a chemical degradation of them. And then you can see if we remove all of the outer seed coat and are left with the inner cuticular part, that matches what we see in the fossils. So that explains why the morphologies are different. And so here, these represent the first fossil records for Cyclanthaceae. Modern Cyclanthaceae is in Central and South America, and here we have fossils in the UK and Germany. So again, not something that you would predict based on where the modern family is at all. The second fossil for mussel that we looked at was another sedge. This one actually is a sedge this time. There's these dispersed individual fruitlets that are widespread in Europe called Caracoidea, and here at Mussel we had these in fructescences, and we actually had some pollen as well. So again, the exceptional preservation meant we could look at these in a lot more detail, and we used SRX-TM again. Since they were quite difficult, we tried embedding them and they just would not section. So I won't bore you with all the details, but basically all the structure of the fruit and the pollen was most similar to this tribe Hypolatry and subfamily Mepanioideae within the Cyperaceae. So that was pretty distinctive. And so this illustrates how fossil taxa can help test some of these hypotheses that people are coming up with based on molecular data. So for example, here's a time tree for Cyperaceae where the stem of Hypolatry was dated to 34.3 million. Here we know this fossil is at 47, so that suggests something needs to be adjusted in the molecular data. Right, and then both of these together, right? So they're not sedges that grow in very open areas. We don't have scirpus, and so this is Cyclenthus here. So this is how Cyclenthus grows, and then the Mepanioid sedges are actually tropical forest understory sedges. So they also grow in this kind of area. So both of these are much more consistent with what the rest of the Mesoliflora says, that these were tropical forests in Europe. We don't have any of these kind of open sedgeland environments. So we're taking this approach now, working with Steve Manchester and colleagues from India to apply this to fossils from the Deccan intertrappean beds from India, which span the Cretaceous-Paleogene boundary. And some of you may have heard a talk by my student Kelly, who talked about one of our first forays into this, which was on this enigmatic monocot Viracarpon. There are two different species, and they have these fruiting structures, but they've been really difficult to interpret. And here we were able to do CT data, and Kelly virtually dissected out, labeled all of the structures. So now we are quite confident in what the actual morphology of the fossil is, and we're working on building a phylogenetic matrix to analyze it, because it's still not clear which family or even order it belongs to. And there have been different hypotheses, Cyclanthaceae, Pandanaceae, Ericaceae, and others have been proposed. So hopefully next year we can tell you what Viracarpon actually is. So for the rest of the talk, I'm going to focus on one of the projects that I'm working on with Chelsea, which is looking at Zingiberales' evolutionary history. And we started working on this group because we thought it was interesting. It's hypothesized to have undergone an ancient rapid radiation. There are very short branch lengths between the families that support that. And we figured, well, if we can really integrate well the fossil and molecular data, maybe we can adjust this and figure out how the characters have evolved, when and where the Zingiberales evolved. And the backbone of the tree, the relationships between the lower families are not so well resolved, so we were hoping by doing this that we could address that as well. So there are 8 families in the order. So we have 4 so-called banana families, which include the birds of paradise, the bananas, the heliconias, and then the orchidanthas. And then we have 4 ginger families, including canna, the spiral gingers, the periplans, and the true gingers, Zingiberaceae. You'll notice these are by far the most diverse in terms of species number. Marantaceae are the second most diverse. So, of course, in order to look at the evolutionary history, one of the things we needed was to actually have a tree with some resolution among the families. And Chodan Sass in Chelsea's lab managed to do this recently. So this came out in PureJ earlier this year, using 308 nuclear and 68 plastid genes from 53 taxa. And we found the banana family, Musaceae, are sister to everything else. And then we recover a monophyletic ginger clade And then we have Heliconiaceae, sister to Loiaceae and Strelitziaceae. So we're happy that we're finally getting somewhere, because now we can move on to some of the next kinds of analyses we want to do, which is integrating the fossils. And so my lab's been busy reevaluating the fossil record, and we've started with we've been working on seeds, and there are quite a few seeds that have been assigned to Zingiberales. Some of them have been placed in extant genera, such as Musa cardiosperma from India, and then Seti oregonensis from Oregon, as well as these couple of European taxa that were placed in the modern genus Alpinia. And then we have some extinct taxa, so a lot of these are placed in the extinct genus Spermatispermum, and then a couple of Mexican fossils in Trigostocarpon and Striatornata. And so our question is really, where do these fossils fit in the phylogeny? So are they in with the gingers? Are they in with the banana groups? Are they something more ancestral than that? Are they stem Zingiberales? And this is showing the geographic distribution, and then the stars are at the level for what age they appear. So we have one species of Spermatispermum that spans from the Eocene through the Pleistocene, so it's quite long-lived. And the oldest fossils within the group are the Spermatispermum chandleri. So we've been focusing a lot of our efforts on Spermatispermum initially, since that'll be key in terms of dating. That's the oldest fossil for the group. So we've reinvestigated 4 species because we could use tomography. We actually have anatomical data from all of these now. I won't bore you with that, but if you're interested, I can show you later. So that was really nice. Some of these only have 2 or 3 specimens, so they're quite rare. And the big problem with Spermatispermum is that it was originally placed in Zingiberaceae in the true gingers, but the most recent paper on it transferred it to Musaceae, to the banana families. And so if you remember, Musaceae is sister or low down on the tree to everything else. Zingiberaceae is in the ginger clades. So if we're trying to use this as a constraint, those are 2 very different positions that will really have a big effect. So we need to place this with more confidence. We need to figure out which family is it actually in. And so to do that, we needed to look at seeds in more detail. Before our study, so the ginger seeds are quite variable in structure. They have a lot of cool characters. And so basically the absence or presence of only 4 characters can let you distinguish between the 8 different families. So that's the aril shown here, the operculum, so this little germination structure, little cap, the micropolar collar, which is a downturning of the seed coat, and then the chalazial chamber, which nobody knows what it does, but it's a little chamber at the chalazial end of the seed. So do seeds have them or not? So we figured we could probably get more than 4 characters out of these seeds. They're highly variable. It's a really fun group to work on. So this is showing you seeds of each of the different families, right? So you can see the arils are all different in these. Look at these arils. Some of them don't have arils, so they're just really cool. And even within the Zingiberaceae themselves, they're super highly variable. So it took a lot more time than we thought to sort these out. And this is largely the work of my postdoc, John Benedict, who's been taking the lead on figuring out the modern seed anatomy. So we've looked at over 200 species using light microscopy. I give a plug for the macropod system that was really helpful in imaging these seeds and helping us see the outside surface of them. And then using the X-ray tomography. And so this was really nice, again, key to be able to study rare things. And so all of this, again, I'm not going to go through everything, but we came up with 50 different seed characters for that. Some of the fun things we can do, since we have these 3D data sets, we can do things like look at the opercula. These are things that would be maybe a millimeter or two in size in real life. And here we can virtually dissect them and look at their morphologies. And this is work a couple of undergrads have contributed a lot to. So just to show you some of them, there actually is a lot of variation in the morphology of just this one structure. And some of them seem to go along family lines. And we can get these out of the fossils too, so that'll help us place the fossils. So with all those 50 characters, we now have a framework that shows us which characters are distinctive for seeds at the family level, as well as sometimes subfamily and tribe. So we can apply that to our fossils. And even within Zingiberaceae, we can distinguish, again, between different clades by combinations of characters. So I can't emphasize this has been a ton of work to do, so John's done a really great job with it. So this has helped us reinvestigate. We looked at these alpinia, so-called alpinia seeds, and I can tell you none of them are alpinia, none of them are Zingiberales. We found some of them were actually sedges, so that same Caracoidea. So they're sedge fruits, and others of them we have no idea what they are. There were at least 3 different taxa hiding within 2 different species of alpinia. None of them are Zingiberales. We could take our seed characters and look at morphospace. So we did an NMES analysis, and this emphasizes each of the families are different. Zingiberaceae occupy a really large portion of the morphospace, which reflects the high morphological disparity that they show. And you'll also note Costaceae and Zingiberaceae and Musaceae are all kind of on this side. That'll come up again. So just to plot the seeds on here, you can see what some of the features that unite all of these. Again, I don't want to go into all the details because we could be here all afternoon talking about them, but I'm sure you guys want to go to lunch instead. So we can use these seed characters to construct. We need to use them to construct a phylogeny with our fossils. We did one with just the extant taxa to understand what was going on. So the big difference is Musaceae actually makes Zingiberaceae polyphyletic, and that's because of some of these shared morphological characters, right? We're not saying that that's the real placement for it, just that there are some features that are found just in those 3 families that are making them do this. If we put in the fossils, so this is an analysis with just the 4 Spirometospermums. They very strongly go in with the subfamily Alpinioideae within the family Zingiberaceae. So they're crown Zingiberaceae. And there's no difference whether we constrain this to be monophyletic Spirometospermum or not with a Templeton test. So we're pretty confident that that's where Spirometospermum goes. John's been doing more analyses with a few of the other fossils, and they also all come up in Alpinioideae. So we've got all of these fossil seeds, and they're all coming into the same subfamily. And there are a bunch of different characters that support this. So we've shown traced down here. One is this character, the micropolar mesotestal proliferation, but we have a bunch of other characters that are supporting the placement of these fossils within Alpinioideae. So where do the fossils fit? So this is the Enseti oregonensis is definitely Musaceae. All of the other seeds all seem to come up into Zingiberaceae. And then, except for the Alpinias, which are not actually Alpinias. So it's kind of weird then that a lot of these fossils are Zingiberaceae, right? If you think back to the morphospace, where the Zingiberaceae has so much more disparity in the seed morphology, that's reflected also in its vegetative morphology and its ecology and the kinds of environments it lives in. And so we're thinking that there's probably, these are probably growing in environments that are much more likely to be preserved, whereas the other families are growing in environments that are much less likely to be preserved, and so we're much less likely to find these in the fossil records. So Zingiberaceae are just, they're buying more lottery tickets for the fossil lottery to get into it. Okay, and so why is this important? So this is some work that Will Isles in Chelsea's lab is doing. He's leading the making the time tree. And what we did was a little experiment just to compare what happens if we put Sparametospermum as a constraint at these different nodes where it's been hypothesized. So if we put it as a stem Musaceae, if we put it as a stem Zingiberaceae, or we put it as stem or crown Alpinioideae. And for a lot of these, I know you can't read the groups, but for a lot of these you can see there's still a large overlap between all of them, even if there's some difference. But I've highlighted here some of the areas where putting it as either stem or crown Alpinioideae makes a huge difference for what the estimation of the node age is. And so this is for the order itself. So we've got about 50 million years difference. And for the banana group, for the ginger group, and then this is for Zingiberaceae proper and Zingiberaceae and Alpinioideae, so the two of the subfamilies. So a really massive difference. So again, this is why it matters, and we need to make sure that the fossils are being interpreted correctly. So you need to be critical about the fossils and where they go. So just for the last little bit of the talk, I wanted to touch briefly on leaves, which is the part of the project that we're getting into. It's not quite as far along, but I'm really excited about this part. It's something I've been wanting to do for a long time, so it's nice to start looking at them. So reproductive structures, we know, have a lot of useful characters for identifying them, which we need for these evolutionary analyses, but a lot of what we find in the fossil record are actually the leaves and not the fruits or seeds, right? And so the leaf architecture working group has already done a ton of work for the dicots with this manual of leaf architecture, which is indispensable, and some recent work, for example, by Peter, has shown we can apply computer vision to finding different characters for dicot leaves, right? We know inherently that we can distinguish between most dicot leaves. We can get a lot out of that. And especially for us paleobotanists, right, we don't always have the fortune of finding whole leaves, we're dealing with fragmentary leaves, so we need to keep that in mind as well. So what about monocot leaves, right? How many people save monocot leaves when they're collecting them as fossils? Yeah, they usually get thrown away or just stuck in a drawer as monocot, right? Because they're all just parallel veined and very boring, right? So there's nothing we can do with them. Well, it turns out they actually have a huge diversity of venation patterns. And so this is what we're excited to be embarking on. So we've got some palms here, there's some elismatales, pandanus, grasses, there's a hosta, there's some other asparagales, Dioscorea and smilax, right? So they're hugely diverse. So I'm convinced that there's more in here, and so I don't know if it's silly or not, but I'll regret it in the end. But we're trying to figure out the leaf architecture, and then that'll give us again a framework that we can use to better interpret the fossil leaves that we have and be able to use those as additional data points. And in a broader sense, I think there's probably some interesting stuff when we're thinking about leaf evolution in general. For example, we know that monocot leaves and dicot leaves develop differently. They develop, and the patterns of vein initiation are different. So, like, what are the homologies between here? We know a lot about how the physiology of dicot leaves and the functions of them, but not as much about monocot leaves, at least not in things like crop grasses. So, and of course that's not saying that no work's been done, because there has been work. There's been work on Araceae, there's been work on a lot of the reticulate veined monocots, especially some nice work by Conover. But again, these are interesting, just showing which families have reticulate venation. They're actually quite widespread. So I think there will be some really neat things to come out of this. What we've been looking at for a start is leaf vein density. So you guys are probably all familiar with this work from Boyce et al., which showed that angiosperms innovated a much higher vein density, so they have higher hydraulic capacity compared to other lineages. And being obsessed with monocots, I was like, well, what are the monocots they have? Most of the values that they have are from grasses, so just from one family. So what's the diversity of vein density there? So to do this, we have measured, so we're going to call it vein length per area because that's more accurate. So we measure the length of all of the parallel veins, and then we measure all the lengths of the fine veins, and then we can add them up for a total VLA. So we're looking at this with three different ways. And again, this has been work by a lot of students, including Molly and Amanda, who are both here, and taking advantage of the New York Botanic Garden and the National Cleared Leaf Collection monocots, as well as ones that we're doing in the lab. So we have a lot more data now. I just haven't finished analyzing it. So I'll show you some stuff. It's still over 100 species of monocots representing 8 orders. So if you look just at average total VLA, monocots are a lot lower than dicots, maybe not surprising. But one of the interesting things is we can plot total VLA against total parallel, just the parallel VLAs, and we see some strong relationships. Here, Poales and Asparagales have a very different, so the slopes are different among different orders, and this is reflecting the structure of the vein. So this is a way to quantify some of the vein architecture. If we look at total VLA versus the fine veins, we these strong relationships for zingiberales, liliales, and dioscoriales. So because of the ginger project, we've been looking at gingers a lot, and we were thinking maybe this is a quantitative way we can take a first pass at trying to figure out what these fossil leaves are. And so we've been reinvestigating some, including this genus Zingiberapsis, this fossil that was placed in the extant genus Canna. This one from India was placed in the extant genus Donax in the Marantaceae. One that was questioned is Costus from the UK. This is an undescribed one from Messel. So where do these go? So if we look just at zingiberales, so again this is total VLA and then just the parallel veins, we see the Zingiberaceae, Strelitziaceae, and Heliconiaceae follow similar trend this way. Musaceae is off on its own. Marantaceae and Costaceae are here. Canaceae is here. Liliaceae is just down here. So they're, again, occupying different parts of the graph, and these are highly significant, statistically significant. And so, again, these are reflecting the different vein architectures. So these guys have a lot of parallel veins and just a few cross veins, so very elongate rectangular areoles. These Costaceae and Marantaceae have much fewer parallel veins. These are all the same area, and much more cross veins. And then the Orchidantha is just kind of weird, so that's why it's down here, right? So if we put in the fossils, then we have Zingiberapsis all falls where we think it should, but here's that Donax that's supposed to be a Marantaceae, so it's not. Here's the Costus is falling within Costus. Here's that undescribed Messel fossil, so that looks like a Marantaceae. Here's where the Canna flaccidifolia falls. It's up with the Heliconiaceae and not at all with the Canaceae. So, like I was saying, I think this is a good way we can do a first pass at this. You can see a lot of differences when you look at the leaves, but they can be hard to articulate in words or characters, and so we needed something a little different. And some of you may know Leo Hickey's barcode approach, and we're trying to apply that, but whenever we've had different people in the lab look at the same leaf and we all come up with a different barcode. So we're trying to figure out how we can refine that and make it a little more repeatable. So this is what we've come up with instead for now. And as I said, it's not like this would be the only thing, so we're looking at other leaf characters that you could then use to confirm belonging in a specific family. So things like the angle of the parallel leaf divergence, whether there's trichomes or not, the shape of the areole, things like free-ending cross veins dichotomizing or anastomosing, all of that kind of stuff. Okay, so I think there's a lot of potential for monocot leaves to give us some new characters, so we can start using that information as well. So just to summarize, reproductive structures have a high number of systematically useful characters, and I showed you a lot from ginger seeds especially, and some sedge fruits, but really that applies to everything. And we've been using a lot of X-ray tomography, and I think it's a really nice technique to use, especially on groups where it can be hard to section their fruits or seeds. It's a very rapid technique, it's non-destructive, and so we can build up these very large data sets of morphology and or anatomy on a decent time scale. And then with other structures, there's lots of potential to better understand them. So I showed you one example from monocot leaves. Of course, there's other parts, stems, rhizomes, and then, of course, this applies to non-monocot groups as well. So dead plants tell no lies, but we can't really hear what they have to say and fully understand what they're telling us and what they're recording without adequately studying both the fossils and the modern material. And to do that, we need a lot of morphology and anatomy. And so once we understand this, we can place our fossils into a phylogenetic context with high confidence, and that's critical for making sure that we're interpreting them correctly, and for all these downstream inferences that depend on that, such as the timing and origin of groups and their biogeography, character evolution, that kind of thing. And so really what we need to do, and I know a lot of us are already doing this, so it's not saying that nobody's doing this, it's just this is such an important thing. We're at this point now where we really need to combine both the fossil and extant data. We need to combine phenomic and genomic data in order to get to the next level of synthesis and understanding. So, thank you.